So the next two presentations are pre-recorded, and so I wanted to tell everyone that and make sure that we have time for them.
Hi, everyone. Thank you for being here today. My name is Jackie Myers. I'm from the Department of Psychiatry and am co-director of the Henry Begleiter Neurodynamics Laboratory. And I have several funding mechanisms from the NIAAA, NIDA, and NIMH, as well as the Brain and Behavior Foundation. And today I'm going to present to you essentially a spin-off of research that Dr. Porches has started presenting in the collaborative study on the genetics of alcoholism study. And I'm going to delve a little bit further into the impact of genomics, neurodevelopment, and traumatic stress on alcohol use disorders. So Dr. Porches very nicely introduced that COGA has around 3 decades of very rich data, including measures of brain functioning. These are measures of EEG, neuropsychological performance, as well as some data on MRI that's been recently collected as part of a spin-off R01 study. And in the next couple slides, I'm going to focus on one of these measures of EEG functional connectivity. For those of you who don't know what this is, I have a little cliff notes on the left-hand side of the slide here. And you could think of this as a measure of communication between neural networks in the brain that occur on a millisecond scale. So using a genome-wide association study approach, in COGA, we ran a GWAS on one of these measures of EEG connectivity, and this is posterior theta EEG coherence. And we saw that one variant on chromosome 18 was really implicated as the most associated with this particular measure in the COGA participants. Interestingly, this top variant influenced the expression of myelin basic protein, which, given the connection between different parts of the brain that is measured in coherence, this is particularly interesting to us. So we then took this variant and looked both in the COGA participants as well as an independent sample, the UK Biobank, and saw that this variant was also associated with measures of drinking, including risky drinking, and symptoms of an alcohol use disorder. And this was both in COGA and in an independent sample. Interestingly, we also looked and saw that the variant was also associated with measures of corpus callosum in the independent sample, and in particular, corpus callosum volume. Finally, we're able to take the longitudinal data that we have in the sample and ask questions about the association between this genetic variant and coherence across development in males and females. And what we could see using this sort of approach is that when we look at the association between individuals from age 12 through around 30, we really see that it's being driven by females in our sample over the age of 25. So really the adult females in our sample are driving this GWAS signal and the associations with alcohol and corpus callosum measures. So in addition to looking at individual variants that are implicated in GWAS studies, we also use a polygenic risk score approach. So here we take these genetic risk scores that are comprised of hundreds or sometimes thousands of individual genetic variants from an external or an independent study. In this example, the Psychiatric Genomics Consortium study of over 500,000 individuals looking at alcohol dependence. We then take that genetic risk score for alcohol dependence and bring it into our COGA study and look at the association between this genetic risk score for alcohol dependence and EEG connectivity across development in males and females in our study. And what I'm showing you is the data here in 2 different ways, the association between these polygenic risk scores and connectivity from ages 12 through 32. And what's at the bottom of the slide are little cartoons of the brain at each developmental stage. And where we see these red lines is really where we see that most robust association between the genetic risk score and our measures of connectivity. So what we can see here is that we really have our strongest effects of this genetic risk score on frontal central and parietal occipital EEG alpha connectivity, particularly in males after age 18, and that's what we're displaying here. And these associations, or these polygenic risk scores, are also associated with increased risk of alcohol use and opioid use disorder symptoms, as well as poorer performance on neuropsychological tasks in the sample. And I've emphasized the data from the males, but just to show you in comparison that we have a very different picture in females. So we're seeing pretty prominent sex differences in our sample and our associations. So we've expanded this work beyond polygenic risk score for alcohol use disorder and substance use disorders to look at other neuropsychiatric conditions. So in collaboration with Dr. Bigdelli and others in the department, we're looking at polygenic risk scores for things like schizophrenia. And here I've kind of show those cartoons of the brain again, just to demonstrate that there are, of course, similarities across associations between polygenic risk for schizophrenia, bipolar, depression, and alcohol dependence, but there are also pretty interesting differences, especially when it comes to looking at sex differences and developmental differences. So for example, in our schizophrenia polygenic risk scores, we saw that the association was most prominent with males between ages 16 and 20, where things like bipolar disorder and depression were observed in both males and females a little bit later in development as well. Finally, we fold in measures of the psychosocial environment. So here I'm showing data looking at the association between childhood sexual trauma and developmental trajectories of one of our measures of event-related oscillations measured during a neural response inhibition task. This is a go-no-go task. And where we see the differences are when participants are expected to inhibit their response, we see this difference in their brain function. And interestingly, this difference that is associated with childhood sexual trauma is in turn associated with increased risk for alcohol use disorders and internalizing disorders, PTSD and depression later in adulthood. We also see associations with poor neuropsychological performance, and these relationships tend to be stronger in females and among those with higher polygenic risk for alcohol use disorders. So the last set of data that I want to show today is data that we've been collecting throughout the COVID pandemic. And like many other samples, we are seeing that there are increases in alcohol and other substance use during the COVID pandemic era. But what's really novel about our study is that we have data that has been, again, collected over 3 decades on individuals prior to the COVID pandemic. So what I'm showing here is that same data on increases in drinking and drunkenness during the pandemic era, but now split as a function of what we knew about their alcohol use disorder status prior to the COVID pandemic. So we're seeing that there are modest increases in those who didn't have a prior alcohol use disorder, but we're really seeing our greatest increases among those that prior to the COVID pandemic had a history of alcohol use disorder, so these 3 groups here, that are then further split into those who had current symptoms, those who didn't quite have symptoms but were risk drinking, and those who were low risk or even abstinent. So we're seeing that even among that abstinent or low risk group, there are increases that have been observed since March 2020. And we were recently awarded a grant from the NIAAA to delve further into this and understand, well, what aspects of the COVID pandemic are really driving these increases in alcohol use and drunkenness. And so far, preliminary data is pointing to measures of perceived stress, decreased social support, and being a frontline worker or an essential worker. So we are also seeing important roles for polygenic risk scores, increasing these associations, and some of those measures of EEG coherence, increasing these associations, and we'll be looking forward to seeing more of these results in the next few years. So I'd just like to highlight some of the ongoing research. Today, I've spoken about the collaborative study in the genetics of alcoholism and several of our spin-off studies and grants. In addition, I've listed here some of our collaborations across Downstate with the Max Wise study and across the country with the NESARC data and the genomic psychiatry cohort. So with that, I'd like to thank, of course, the scientists that were very helpful in collecting and analyzing a lot of the data that I showed here today, as well as our COGA collaborators and participants, and our funding from the NIH and Brain and Behavior Research Foundation, and of course, Downstate and the organizers for today's symposium. So thank you.